as you know, the main pathogenic event in prion diseases, independently of how they are acquired, is the conversion of the cellular, of the cellular form of the prion protein into this abnormal form, which is called PRPSC, which tends to form insoluble aggregates. And this form of the protein is able to convert the normal form of the protein into a likeness of itself. So this is a schematic of this process where PRP scrapie is indicated by these purple diamonds and PRPC by this round circle, blue circle. So when aggregates of PRP scrapie get in contact with PRPC, they induce it to convert and to be recruited into these aggregates. So the aggregates grow, and then what we think is that cellular processes break down these aggregates in new, smaller aggregates, which we call seeds, which can promote this misfolding process. So since this is the central event in prion diseases, it has been the main target for therapy. And you can think of acting on this process at different stages. One that we have just heard about is to downregulate the level of the cellular prion protein, and there is proof of principle that this can be done safely and that it is a good therapeutic approach in prion diseases, at least in the models. You can also think of stabilizing the PRPC conformation so that, and that in principle can be done by finding small molecules that bind to PRPC and that lock it in its native, in its normal conformation so that the protein cannot be converted into PRP scrapie, or at least is less prone to undergo conversion. But you can also target other steps of this replication process. So a physical interaction between PRP scree and PRP scrapie and PRPC is required for the conversion of PRPC. So you can think of molecules that block this interaction or that inhibit the elongation, the formation of polymers, or their fragmentation. Or you can also think of treatments that promote disaggregation and degradation of PRP scrapie. And for instance, we know that one of the cellular processes that is involved in clearing prions from cells is autophagy, and there are drugs that can improve autophagy, can boost autophagy in the cells. So we are working on a molecule, which I will show you in a minute, which we think acts on two different steps of this replication process. I won't go into the details of how the mechanism of this molecule is, but I will tell you what it does to models of prion disease. So this is the molecule. It's a porphyrin, which we call the VA01. This is just the core of the protein, of the molecule. I cannot show you the full structure because it's potentially subject to a patent, so I was asked not to show you the full thing, but I can tell you that it's very similar to these other porphyrin. The difference is that this one has iron in the middle, ours has zinc, and also these lateral groups, which are these R here, are slightly different. This one was described in the late 90s by Byron Coy, whom you just heard, and it has been shown to have an antiprion activity and to be a potent antiprion molecule. So in the experiments that I'm going to show you in a moment, we will always compare our molecule with this gold standard. So in the first set of experiments, we used the PMCA, which is protein misfolding cyclic amplification, which you have heard about it yesterday. This is a technique which allows you to amplify prions in a test tube. It's a cell-free system in which you have an excess of cellular prion protein and a small seed of PRP scrapie. These are incubated together so that the PRP scrapie recruits PRPC and alongs, form these polymers, which are then broken down by sonication, which are sound waves, to produce more seeds, and the cycle is repeated. And you can amplify PRP scraping indefinitely with this system. This is used for diagnostic purposes, but can also be used to test antiprion compounds which inhibit this process of prion replication. So when we did this experiment, you can see here, this is the amount of seed that we used, and this is the control in which the amplification in control conditions. And you have a approximately 8-9-fold increase in the amount of PRP scrapie with a single round of PMCA. If you do this experiment in the presence of VA01, and these are 3 independent test tubes, you see that this process is strongly inhibited, and this is the quantification. And the same holds true when you use the iron porphyrin described before. Then another kind of assay that we used is a cell assay. These are neuroblastoma cells which are chronically infected with prions, with a 22L strain of prions. And these cells propagate prions chronically. And how do you test that? You can take these cells, lyse them, and then subject the lysate to proteinase K digestion. While in non-infected cells, PRPC is completely degraded by proteinase K, PRP scrapie is partially resistant to proteinase K digestion. So after you do that, and you digest with PK, you can see these 3 bands which corresponds to the PK-resistant core of PRP scrapie. So you can treat these cells with your compound of interest and see whether the amounts of PRP scrapie decrease. This is exactly what we did with our molecule, and we used different concentrations of VA01 in parallel with the iron porphyrin. And this is the control condition. This is as well is a control condition with no compound, and you can see the PK-resistant fragments here. Then cells have been treated with these different concentrations of the compounds for 1 week, and what you can see is that PRP scrapie decreases and completely disappears when you go up with the concentrations. And we know that these cells have been completely cured from infection. They do not propagate prions anymore. With this compound, you have a less strong effect, and we repeated this experiment several times, and we can conclude that our porphyrin is twice as potent as these other porphyrin. Another system that we used is the prion-infected cerebellar organotypic cultures, which is another model, in vitro model, which is closer to the brain. In this experiment, you take the brain of mice and you dissect the cerebellum. The cerebellum is then cut into slices. The slices are exposed to prion, to a prion-infected brain homogenate from mice, and for 1 hour, and then they are put in Petri dish and cultured for several weeks. And what happens is that during this period, prions replicates and propagate in the tissue, and so you can see that by treating the slices with proteinase K again, and this is the results of the experiment after 1, 3, or 5 weeks in culture, and you see that the amount of PRP scrapie increases. This is the control that has been exposed to a normal brain homogenate which does not contain prion and is not infected. So again, you can take these organotypic cerebellar cultures and infect them, let them go for a while, and then treat them with your compound of interest. And when we did this experiment with VA01, you see that compared to the control, where you can detect PRP scrapie, the amount of PRP scrapie is strongly reduced, much more than in cultures treated with the iron porphyrin, which was active, a little bit active, only at the highest concentration. So from the previous experiment and this experiment, we can conclude that our porphyrin is a good antiprion molecule with a potent activity. The problem with these molecules, with porphyrins in general, is that they do not cross the blood-brain barrier efficiently. They don't reach the brain very efficiently. So we ask whether our porphyrin cross the brain and reach the brain. So to do that, we did pharmacokinetics studies in which you give the molecule to the periphery of mice in this case. We injected it IP intraperitoneally, and then we measured the amount in the blood, in the plasma, or in the brain. And after half an hour, 1 hour, you have a peak of compound in the plasma that then goes down because the compound gets into the organs and part of it is degraded. And some of it could also be detected in the brain after 2 hours, a peak after 2 hours, and goes down in a slow fashion. So, of course, this is much less than what you get in the plasma, and is much less than the active concentrations in the experiments that I showed you before. But since it is slow degraded from the brain, we thought that we could chronically give it to mice in order to reach a higher level in the brain. And in fact, if you give it for 4 weeks, 3 times a week, you reach a steady state level in the brain, which is more than 100 nanograms per gram, which is approximately 1 micromolar, which is approaching the concentrations which were active in the in vitro systems. So we treated the mice, and we got some preliminary encouraging results because it seemed to be very active in the brain, but then we repeated the experiment 2 more times, and the results were disappointing. So we think that we are reaching a threshold concentrations in the brain, which is just not enough to have a consistent biological effect, curative effect. So how can we boost the brain delivery of this molecule? One possibility is to use nanoparticles, and we chose to use nanoparticles made of polylactic glycolic acid, or PLGA. This is a polymer which is not toxic, is safe. It has been approved by FDA for several uses. It is relatively inexpensive and can be modified chemically, and this is important as I show you in a moment. These PLGA can form nanoparticles, which are very small objects. They are 100 to 200 nanometers in diameters, and these are spherical and can be loaded with various molecules. So we thought that we could load our molecule into these nanoparticles and use these as vectors to bring them to the brain. However, PLGA nanoparticles, as they are, do not go efficiently to the brain. You have to modify them. And one way of modifying them to reach the brain is to attach on their surface this small peptide, which is a sort of a little protein. It's a 7 amino acid glycopeptide, which is like an opioid-like glycopeptide, which is modified to avoid the opioid effect. But it is recognized by opioid receptors, so it can be taken up by cells, and because of that, it can cross the blood-brain barrier and get into the brain. And there is evidence that these nanoparticles, loaded with different compounds, can reach the brain and have curative effects in mouse models of Huntington disease, and as well as Alzheimer's disease mouse models, and many others. These are just 2 examples. This is the reason why we chose to use these kind of nanoparticles. So in a first experiment, we loaded VA01 into these nanoparticles. First, we wanted to know whether that could be efficiently loaded in nanoparticles. And we could do that. And then we did some preliminary experiment. This is just an example. So in this case, we used the prion-infected cocks, the cerebellar organotypic cultures, which were either untreated or treated with the empty nanoparticles, or with nanoparticles loaded with the VA01, or with the free, or treated with the free VA01. And as you can see, this is just one single treatment, and the cocks have been analyzed after 48 hours. So it's different from the experiment that I showed you before, in which cocks had been treated for 1 week. So the effect is smaller, but you can see that VA01 is active as we expected, and the VA loaded in nanoparticles is active as well. It reduces the amount of PRP scraping, whereas the empty nanoparticles don't do anything. So this is very promising, and this is the stage where we are now. We are going to now test whether we can use these vectors, nanovectors, to bring VA01 to the brain of mice. So in summary, VA01 inhibits PRP scraping replication in PMCA system in prion-infected N2A cells and COX. Is more potent than the iron porphyrin described before. A fraction of the molecule reaches the brain after systemic administration, but its biological activity in the brain is variable, so we needed to improve its delivery to the brain, and nanoparticles, functionalized nanoparticle carrying this G7 peptide can improve brain delivery of drugs. That has been shown in other diseases, in other models, and we have now shown that VA01 can be efficiently loaded in G7 nanoparticles and maintain their antiprion activity in COX. So in conclusion, this is a promising, we think, is a promising therapeutic molecule for prion diseases. But before testing its therapeutic activity in preclinical models, we have several preclinical models. There are many models in the field of both acquired and genetic prion diseases, which we could use. We needed to improve its brain penetration, and we have shown that it can be loaded in nanoparticles and maintain its activity in vitro. And so the next step, as I said, is to test the brain delivery of VA01 loaded in nanoparticles. So with that, I conclude, and these are the people who collaborated in my lab, in particular Antonio Masone, Giada Lavinia, Elena Restelli, and former lab members. The group of Marco Gobbi did the pharmacokinetic studies. We are collaborating with the University of Modena, Giovanni Tosi, for the nanoparticle work, and Stefano Banfi and Enrico Caruso are the chemists who produced this molecule, the VA01. Romolo Nonno and Ilaria Vanni did the PMCA experiments, and we're also collaborating with other groups at San Raffaele Hospital and with Jesus Requena at University of Santiago de Compostela in Spain for part of the project, which I didn't have time to show you. But this is the most important slide because it lists the support, the important support of the Creutzfeldt-Jakob Disease Foundation, in particular the Cheryl Molloy Memorial Research Grant contributed by Tim Molloy, whom I had the pleasure to meet here, and his family, who entirely funded my grant. And this project is also funded by the Telethon Foundation and by the Italian Ministry of Health. And I want to thank you for your attention.